They say nature hides her greatest cures inside silence, in the pigments of a fruit, in the bitter edge of a seed, in molecules that seem too humble to matter. Among these, a group of compounds once thought to be simple antioxidants has begun to reveal a deeper intelligence. These are the polyphenols, tiny guardians scattered across the skin of grapes, the veins of olives, the flesh of berries. In laboratories, they whisper to human cells, influencing the very language of life itself, DNA. Some researchers now describe them as molecular readers, capable of touching the code of a tumor and altering its fate. Few people understand how profound this discovery could become. It is not just chemistry, that it is a conversation between nature and our own biology, a forgotten dialogue that modern science is finally decoding. Stay with me. This is one of our short documentary explorations into the unseen world of medicine. If you enjoy discovering how the body and nature interact in ways rarely explained, like this video and subscribe to the channel. Remember, this is not medical advice. It's a journey of understanding, not a prescription. Long before we had words like antioxidant or epigenetic, humankind already sensed the hidden wisdom that ripened beneath the sun. The crimson hue of a pomegranate, the deep blue of a grape, the golden shimmer of an olive, all carried an aura of protection, a sacred vitality that healers revered without understanding its chemical heart. In Egypt, they crushed berries into potions to preserve youth. In ancient Greece, physicians recommended olive oil for resilience and balance. In the Far East, tea was a ritual of clarity, not merely a drink. Across civilizations, the bitter, colorful, and aromatic were trusted as life-affirming, long before we could measure molecules or observe cells dividing under a lens. When the 19th century brought the microscope and the spectrometer, curiosity turned myth into method. Chemists discovered strange crystalline substances in plant extracts that resisted oxidation, the same invisible force that slowly ages both fruit and flesh. They named them polyphenols, referring to their multiple phenolic rings capable of trapping reactive oxygen species. Yet even that early description was incomplete. Over the following decades, as cellular biology unfolded, scientists realized these structures were not inert. They were dynamic communicators, molecular diplomats mediating conversations between the plant world and our own physiology. In laboratories, they began to affect enzymes that regulate inflammation and cell growth. They could neutralize free radicals, bind to DNA, and influence how genes were expressed. It was as if plants had been preparing us, molecule by molecule, for survival. The 20th century brought patterns that medicine could not ignore. Epidemiologists studying the French paradox found that populations consuming red wine and olive oil had fewer heart and cancer deaths, despite diets rich in fats. Researchers traced this protection back to polyphenols like resveratrol, hydroxytyrosol, and quercetin. Parallel studies in Japan linked green tea catechins to reduce tumor formation. Even cocoa, once dismissed as indulgence, revealed complex phenolic compounds that modulated vascular and cellular health. Suddenly, nutrition became biochemistry, and culture became evidence. The quiet rituals of eating and drinking turned into experiments of molecular consequence. By the early 21st century, this scientific awakening had deepened. The discovery that polyphenols could interact directly with DNA and influence epigenetic behavior changed everything. They were not just shields against damage. They were editors of biological memory. These molecules could silence oncogenes, reactivate tumor suppressor genes, and reprogram cell destiny through pathways involving DNA methyltransferases, histone deacetylases, and microRNAs. The same molecules that once gave flavor and color to a fruit could also rewrite the language of life inside a cancer cell. It was no longer folklore, no longer coincidence. It was evidence of an ancient alliance, nature's quiet defense, waiting to be rediscovered by modern science. To understand how fruit polyphenols communicate with tumor DNA, we must descend into the molecular underworld, the hidden corridors of the cell where light and shadow coexist. DNA, the keeper of identity, is constantly threatened by oxidative stress, mutations, and misfolded instructions. Cancer emerges when that order collapses, when the language of replication becomes chaotic. Polyphenols seem to recognize this chaos not as enemies but as editors gently correcting a corrupted text. Their structures, aromatic rings adorned with hydroxyl groups, allow them to bind selectively to DNA and its regulatory proteins, influencing how the double helix breathes, twists, and transcribes. In controlled studies, researchers have observed how compounds like resveratrol from grapes, epigallocatechin gallate from green tea, and hydroxytyrosol from olives can interfere with DNA methyltransferases, the enzymes that lock genes into silence. When these enzymes are restrained, suppressed tumor suppressor genes can awaken again, restoring some order in the genetic script, but their influence stretches far beyond methylation. Polyphenols can also remodel chromatin by inhibiting histone deacetylases, freeing DNA that had been tightly wrapped and inaccessible. They alter the choreography of transcription factors like NF-κB, p53, and STAT3, which decide whether a cell should repair itself or self-destruct. In tumor cells, this translates into the slowing of uncontrolled proliferation and the reactivation of apoptosis, the graceful death that keeps balance in living systems. It is almost poetic. Molecules born to defend a fruit from ultraviolet damage and microbial invasion now defend human cells from internal disorder. The same resilience that lets a berry survive the sun becomes a blueprint for cellular healing. Laboratory studies have gone even further, revealing direct physical interactions between certain polyphenols and DNA strands. Advanced spectroscopy techniques have shown that quercetin, genistein, and curcumin can intercalate within the DNA helix, sliding between base pairs, slightly altering its flexibility and transcriptional accessibility. Though delicate, these changes can determine whether a malignant gene remains active or dormant. Scientists describe this as nutritional epigenetics, the way food molecules become co-authors of our genome's story. The body doesn't simply digest them, it reads them. The most fascinating element of this discovery is coordination. Polyphenols rarely act alone. Inside the cell, they form a molecular orchestra, each compound amplifying or stabilizing another's function. For example, resveratrol enhances the bioactivity of quercetin, while catechins from green tea synergize with curcumin to suppress metastatic signaling. Some even influence microRNAs, the subtle messengers that regulate entire networks of genes. Through them, polyphenols can quiet pathways that lead to invasion and angiogenesis, depriving tumors of their blood supply. What is most humbling is how small doses, sustained over time, appear to reshape biology more profoundly than any synthetic intervention alone. Not through violence, but through persuasion, guiding cells toward harmony instead of forcing them into submission. The fruit, it seems, was never just food. It was a biological conversation waiting to be heard. Among the many studies exploring this molecular dialogue, one stands out for its precision, the research published in MDPI's Oncology and Therapy journal, revealing how olive oil polyphenols interact with tumor DNA. The scientists behind this investigation examined compounds like hydroxytyrosol and oleuropein, tracing how they could modulate gene expression linked to carcinogenesis. Their data showed that these natural molecules didn't merely block oxidation, they actively communicated with epigenetic machinery. Hydroxytyrosol, for instance, was shown to reduce DNA damage markers and modulate the expression of p53, a master regulator of cellular life and death. Oleuropein in turn influenced apoptosis through the Caspase cascade and interfered with inflammatory transcription factors like NF-κB. Together, they demonstrated that components of something as ordinary as fruit oil could exert a molecular sophistication that rivals synthetic drugs. The concept of nutritional genomics suddenly became tangible, food as a tool of genetic regulation, not just energy. The researchers went further, noting that polyphenols operate through both direct and indirect mechanisms. Directly, they interact with nucleic acids and DNA-binding proteins, altering the chemical accessibility of chromatin. Indirectly, they modulate redox-sensitive signaling pathways, enhancing the expression of antioxidant enzymes such as superoxide dismutase and catalase. This duality, chemical precision combined with systemic harmony, is what makes them so extraordinary. It also helps explain why diets rich in polyphenols often correlate with lower cancer incidence, particularly in populations adhering to Mediterranean-style eating patterns. These effects are not merely correlations. The molecular evidence is mounting to suggest causation. The olive tree, long revered as a symbol of peace and endurance, is revealing another layer of meaning, a biological resilience that humans can borrow through nutrition. Yet, the study also confronted the limits of nature's power. Polyphenols, though potent in vitro, struggle within the human body to maintain stability and reach effective concentrations in tissues. The intestinal microbiome, liver metabolism, and rapid clearance often reduce their bioavailability. The researchers proposed advanced delivery systems, nanoparticles, liposomes, and polymer conjugates to overcome these barriers. Such innovations blur the boundary between food and medicine, transforming an ancient oil into a biotechnological agent. The idea that nutrition can influence DNA through engineered natural molecules hints at a new frontier, personalized nutrigenomics. Still, the tone of the research remained cautious. The scientists acknowledged that while cell culture and animal models offer compelling data, human trials are scarce and complex. The molecular intimacy between polyphenols and DNA cannot yet be fully controlled or predicted inside a living organism. Variables like age, diet, and genetics alter the outcome. However, what the MDPI paper made clear is that the relationship between fruit polyphenols and tumor DNA is not speculative. It is measurable, reproducible, and increasingly undeniable. We are witnessing a shift in how we perceive natural compounds, not as weak imitations of drugs, but as ancient molecules carrying evolutionary memory, codes written by plants and interpreted by our cells. What was once dismissed as poetic romanticism is now quantified through spectroscopy, gene sequencing, and computational modeling. The olive branch, once a symbol of peace between nations, has become a molecular symbol of peace within, between human biology and the natural intelligence it evolved alongside. In this reconciliation lies the quiet promise of future medicine, therapies that don't conquer nature, but collaborate with it. The relationship between polyphenols and the pharmaceutical industry is, at its core, a paradox. On one side, these compounds inspire awe for their complexity and elegance, molecules capable of guiding DNA behavior through non-invasive, naturally occurring chemistry. On the other side, they present a commercial problem. They cannot easily be patented. No company can truly own a molecule that grows in a fruit or a leaf. For that reason, large pharmaceutical enterprises have often approached polyphenols with cautious distance, preferring to synthesize derivatives that can be patented, standardized, and sold as drugs. This has created two worlds of research, one nourished by curiosity and human health, the other driven by profit and regulation. Yet even as industry hesitates, laboratories continue to publish compelling evidence that these natural compounds interact with cancer biology on a level of subtlety that many synthetic agents cannot achieve. Polyphenols such as curcumin, resveratrol, and epigallocatechin gallate have all been tested in cancer models, showing the ability to suppress oncogenic signaling, restore tumor suppressor genes, and enhance the sensitivity of malignant cells to chemotherapy. But their bioavailability, the fraction that actually reaches the bloodstream and tissues, remains the central challenge. Curcumin, for example, exhibits remarkable anti-tumor activity in vitro, but is rapidly metabolized in the human liver. To address this, researchers have developed formulations that attach polyphenols to nanoparticles or liposomes, which protect them from degradation and allow a slow, targeted release into tissues. In these studies, industry and academia meet halfway. The natural molecule provides the intelligence, while biotechnology provides the vehicle. However, this collaboration also reveals a darker undertone. Many natural compounds lose their harmony when isolated or over-refined. When pharmaceutical companies create synthetic analogs, the resulting molecules can act more aggressively than their natural counterparts, sometimes disrupting the very balance that made them beneficial. For instance, certain flavonoid derivatives designed for enhanced potency have demonstrated cytotoxic effects on healthy cells, undermining the purpose of their creation. The wisdom of the original molecule, its gentle negotiation with biology, becomes lost in translation. This is the irony of modern medicine. In the pursuit of control, we sometimes silence the intelligence of nature itself. Meanwhile, the supplement industry has taken a different path, commercializing polyphenols as capsules and powders. Often without standardized content or bioactivity verification, consumers drawn to the idea of natural healing flood the market with demand. Yet most products lack rigorous testing. Some contain only traces of the compounds advertised. Others combine polyphenols with additives that hinder their absorption. This chaotic marketplace stands in contrast to the meticulous studies being done in academic institutions. The gulf between science and commerce widens, leaving the public unsure whom to trust. Still, progress continues. Pharmaceutical innovators are beginning to explore semi-synthetic hybrids that preserve the natural core of polyphenols while improving stability and bioavailability. Companies in Japan, Italy, and the United States are experimenting with encapsulated resveratrol, hydroxytyrosol, and EGCG for adjunct cancer therapy. The results are promising, but still early. These trials reveal a subtle truth. The medicine of the future may not belong exclusively to the laboratory or the forest, but to the space in between. Polyphenols may guide humanity toward a new therapeutic philosophy, one where nature is not merely a resource to exploit, but a partner to collaborate with. The MDPI study and its successors represent this philosophy taking form. They do not claim miracles. They reveal interactions, delicate, verifiable, and profoundly human. The pharmaceutical industry, once dismissive of such natural complexity, is beginning to recognize that evolution itself may have crafted the most refined molecular engineers, plants. The question now is not whether polyphenols work, but how to respect their language without rewriting it beyond. Recognition, understanding how to use polyphenols, wisely begins with recognizing that they are not medicines in the conventional sense. They are participants in our daily dialogue with biology. Their power is cumulative, not immediate. The body listens to them slowly, through meals, through seasons, through the quiet persistence of routine nourishment. The richest sources are scattered throughout nature's palette: the violet skins of grapes, the deep reds of berries, the golden sheen of olive oil, the earthy tones of cacao and tea leaves. Each contains its own constellation of polyphenols, resveratrol, anthocyanins, quercetin, catechins, and phenolic acids, each speaking a slightly different dialect of cellular language. What unites them is their ability to modulate the oxidative and inflammatory balance that defines health. In practical terms, diets emphasizing variety and freshness yield the greatest benefit. Studies show that a Mediterranean pattern, abundant in fruits, vegetables, legumes, whole grains, olive oil, and moderate wine, sustains higher plasma levels of polyphenols and lowers systemic inflammation markers such as C-reactive protein. Similarly, Asian dietary traditions rich in green tea, soy, turmeric, and seaweed provide a unique portfolio of compounds with complementary activity. Yet the secret lies not in excess, but in continuity. Unlike pharmaceuticals, polyphenols demand constancy rather than concentration. A daily glass of red grape juice or a drizzle of cold-pressed olive oil influences cellular signaling more effectively than an occasional high-dose supplement. The body thrives on rhythm, molecules entering not as invaders but as familiar voices reminding our systems of their natural cadence. For those seeking concentrated forms, extracts and supplements can play a supportive role, though they must be chosen carefully. Standardization is essential. Polyphenol content should be verified through scientific analysis rather than marketing claims. Products containing standardized amounts of EGCG from green tea, resveratrol from grapes, or hydroxytyrosol from olive oil offer measurable activity. But they are still adjuncts to, not replacements for, balanced nutrition. Timing also matters. Consuming polyphenol-rich foods alongside fats enhances absorption, as many of these molecules are lipophilic. Conversely, overconsumption of alcohol or processed sugars can diminish their efficacy. As these substances compete for similar metabolic pathways, there is also a fascinating role for the gut microbiome, the vast ecosystem that interprets many polyphenols into bioactive metabolites. When we consume them, only a small portion is absorbed directly. The rest travels to the colon, where bacteria transform them into simpler compounds capable of crossing into circulation. This process varies from person to person, which means that two individuals consuming the same food may experience entirely different molecular outcomes. Scientists call this nutritional individuality. It is here that the future of personalized medicine may blossom, tailoring diets to the microbial and genetic profiles that determine how our bodies translate the language of polyphenols. And yet, humility remains crucial. These compounds are potent, but they are not magic. They complement medical therapy. They do not replace it. Their ability to support DNA repair, reduce inflammation, or sensitize tumor cells to treatment makes them allies, not alternatives, to modern oncology. For those battling cancer or chronic illness, introducing polyphenol-rich foods under professional guidance can support resilience and recovery, but abandoning conventional treatment is never advised. The key lies in integration, the meeting point between clinical science and ancestral wisdom. When consumed with awareness and respect, polyphenols become more than nutrients. They become teachers, reminding the human body of its ancient alliance with the plant world, a partnership that has always whispered of survival, balance, and renewal. The story of polyphenols and cancer is far from finished. In truth, it may only be beginning. What current science reveals is the outline of a much greater design, a biological architecture in which natural compounds and human DNA are not strangers, but long-separated relatives rediscovering each other. The next decades will determine how we translate that revelation into practice. Research is already expanding into precision nutrition, where genetic and epigenetic mapping will help identify which combinations of polyphenols work best for specific individuals and tumor types. Some teams are developing nutripharmaceuticals, tailored compounds derived from fruits that target particular oncogenic pathways. Others are experimenting with artificial intelligence models that predict how plant molecules will bind to cancer-related genes before any experiment begins. Slowly, a field once dismissed as folk wisdom is merging with quantum chemistry, bioinformatics, and nanomedicine. The most promising frontier lies in delivery systems. Scientists are designing nano-encapsulated polyphenols capable of bypassing the gut's destructive enzymes, traveling intact into the bloodstream and releasing their contents only when they reach malignant tissue. These nanocarriers, often coated with biodegradable polymers, can even be guided by magnetic or pH-sensitive signals. In animal models, such systems have enhanced the stability of curcumin, the permeability of EGCG, and the potency of resveratrol by several orders of magnitude. In parallel, research into polymeric hydrogels and bioliposomes suggests that we may soon administer these compounds through slow-release implants or intravenous infusions, turning what was once a glass of juice into a precision therapy. The concept sounds futuristic, but it grows directly from nature's own logic: gradual release, precise targeting, minimal harm. At the same time, a philosophical transformation is underway. The medical community is beginning to view food not only as fuel, but as molecular information, a language that instructs cells in real time. Polyphenols are among the clearest expressions of this truth. They show us that nutrition and genetics are not separate domains, but chapters of the same evolutionary manuscript. When we eat, we do not simply consume matter. We communicate. The body reads every molecule, interprets every signal, and responds with intricate feedback loops that shape its resilience or vulnerability. Cancer, then, can be seen not merely as a disease of mutation, but as a breakdown of this molecular communication, a dialogue interrupted by excess, deficiency, or dissonance. Still, caution must accompany wonder. Even the most elegant natural molecules have limits. Some polyphenols exhibit biphasic effects, protective in low doses but potentially pro-oxidant at high concentrations. Others can interfere with certain medications or hormonal pathways. This complexity demands not fear but respect. It is a reminder that nature's chemistry is balanced by boundaries and that wisdom lies not in consuming more, but in listening better. Future clinical trials, rigorous, transparent, and inclusive, will be essential to define safe and effective uses. If successful, they may usher in a form of medicine that transcends the old division between natural and synthetic, uniting them in a continuum of biointelligence. For now, the simplest act remains the most profound: to nourish oneself with color, diversity, and patience. Each fruit, each leaf, each drop of oil carries messages millions of years old, instructions written in chlorophyll and sunlight awaiting translation in our cells. Perhaps this is the quiet revolution of polyphenols, not an attack on disease, but a restoration of conversation. The human body was never meant to fight alone. It was designed to cooperate with the living world. In that cooperation lies hope, not the fragile hope of miracle cures, but the grounded faith that life, when aligned with nature's code, tends to protect its own. Stay curious, my friend. The next discoveries will not only tell us what polyphenols do, but what they remind us of. That healing may not always require invention. Sometimes it requires remembering